So we are in the anatomy of a microscope, and I'm going to share my screen now. Sorry for being a tiny bit late. Yes, here, or how do I do that? Here. Look, that's it. And hide, and that's it. So today we're going to talk hardware. So microscopy is a very vast field. Within microscopy, there's lots of different branches, if you want to say. So we have material imaging, for example, which is a huge branch when it comes to money. And then we have the scanning probe microscopy is a tiny niche. And we have electron microscopy, which we know very well, transmission or scanning. And we also have, of course, medical imaging, which is a huge branch when it comes to money. And then we have our little niche, which is light microscopy for biological research. And that's what we are talking about in this course, and only this. So within this field, we can image basically samples in two different ways. We can use transmitted light microscopy. There's a few more ways, okay? But in this course, I'm focusing on the two main ways. So transmitted light microscopy, one of the main type of techniques that we use is the bright field techniques, and I will talk about that and a few more. So transmitted light microscopy or fluorescence microscopy, right? So in transmitted light microscopy, we can use a sample that is labeled, for example, with eosine hematoxylin, or it can be a sample that is label-free. In fluorescence microscopy, we can also have a sample labeled or labeled-free. For example, if you have an autofluorescent sample, you may use it without labeling it. You can also use reflection, for example. Diffraction microscopy is something where we image the laser light that reflects on the sample. And there are several other ways to image a sample in fluorescence without labeling it. So we can have label and label-free in these two types of microscopy. And then transmitted light microscopy can be acquired on a wide field system or on confocal microscopes. And it's the same thing for fluorescence microscopy. We can have it on wide field systems, on confocal microscope, but also on light sheet systems. So these two things, transmitted light microscopy and fluorescence microscopy, are different microscopy techniques. These here are wide field confocal and light sheet. They are different types of microscopes, and you will see later why I prefer to call them systems. And then we also have on each system, we have the microscope stand, like the microscope, the one you look at in the eyepiece, the one that has the eyepiece and the stage and everything. And sometimes they are called the microscope body as well. And these can be upright, inverted, or flat. So I will show you all this today. And these are different types of microscope stand architectures. So it can be a little bit confusing, let's say, but I think at the end of this day, you will know exactly the difference between transmitted light microscopy, fluorescence microscopy, and this type of microscope, and also these guys. So we're going to be working on this now. So one thing that I really want to point out, because it's super confusing, is that we have bright field and wide field, and these things are completely different. So it's easy to mix them up, but actually it's logical. So if you see a bright field, means that when you look at the image that were acquired in bright field microscopy, the objects are dark. So your cells will be dark objects on a bright background. So the background is bright and the objects are dark. This is bright field. This is an image acquired with bright field microscopy. On the other hand, something completely different is a type of microscope, and it's called wide field microscope. And this means that the illumination, the excitation light, is defocused. And this is why it's called wide, because it's not focused. Okay, so these two things have almost the same name, but they're actually very different. This is a type of microscopy technique, and this is a type of system. So what is the difference between all this transmitted light, fluorescence, and the different types of microscopes and all this? It is the light path. So we're going to talk a lot about light path today. So understanding the light path always starts with the light source. And then we have the optical elements, and then we have the sensor. So it always starts with the light source and it ends with the sensor. And in between, lots of optical elements. So let's define some things, and we're going to be using some words throughout the course in such a way that we talk about the same thing. And then we can, discuss, is sensor the right word or not the right word? Doesn't matter. What matters is that we define some words that we're going to use throughout the course. So when I say sensor in this course, I mean the light recording device. So can anyone tell me what type of light recording device we can have on the microscope?
A camera,
Camera, for example, is a light recording device. Absolutely. What else?
A detector.
What was that?
A detector.
Detectors are another type of light recording device. Absolutely. There's another one. Maybe I don't quiz you, huh? It's the eyes. If we are actually looking in the eyepiece, we're using our eyes as light recording devices. But you're absolutely right. So eyes, detectors, and cameras. And you will see that there's a fundamental difference between detectors and cameras. So which optical elements are we talking about here? It can be all the lenses, including the objective, the lens of the objective. So they are optical elements in the light path. But we also have the immersion medium of the objective. So whatever the oil that we put on the oil objective, or the water we put, or air, is also some sort of a medium that is on the lens. Okay, all this is part of the optical elements. But we also have all the filters. So what type of filters could we have in a microscope?
Excitation filter and emission filter.
Yes, we can have excitation and emission filters, exactly. And so the excitation filters, they are always close to the light source because they deal with the excitation. So it's a little bit logical that they are just close to the light source. And the emission filters, they deal with the emitted light that is going to be recorded. So the emission filters, they are always very close to the sensor. Okay, so this is sort of logical where they are in the microscope. And then we have, of course, dichroic mirrors. So please shoot me. What are the four roles of a dichroic mirror? Anyone?
directing the excitation towards the sample and
Yes.
the excitation laser away from the sensor.
Yes. That's
And,
two.
and then I guess the emission should be transmitted to the sensor.
The emission has to go through the sensor.
And I don't know if this is the fourth, but splitting the excitation and the emission paths or lights.
Okay, so you have the first three basically are giving the four. So the first three roles that you said, sending the excitation to the sample, sending the emission to the sensor, and preventing the excitation from going to the sensor, these are separating the excitation from the emission. But the fourth one is that sometimes we use dichroic mirrors to separate two emitted lights. So when we have two different sensors and we would like to send, for example, the green light to one sensor and the red light to another sensor, then we can use a dichroic mirror also to separate two different types of emission. So this would be the fourth one, right? So first we send the excitation light towards the sample, logical, right? And then we send the emission light towards the sensor. Clearly, this is what we want to record. And then we prevent the reflected excitation light, the excitation light that reflects on the sample that we don't want it to reach the sensor. So we prevent it from doing that with the dichroic mirror. And then the last one is that sometimes we can use a dichroic mirror to split two emitted lights between two different sensors. So well done. So here are the dichroic mirrors. They're basically going to be between the excitation light and the emitted light because this is where they split. It works a little bit like a roundabout, the dichroic mirrors. You go here, you go here. All right. So in the light path, another type of optical element is pinholes. And these are only for controcals. So we'll talk more about pinholes today. But it's important to realize that the sample is also in the light path. So the sample is part of the design of the microscope. When the microscope manufacturers design their microscope, they are assuming some things about your sample, and it's important that you prepare your sample in that way. I'm going to just have a quick look here because I want to keep track of time nicely. Yes, okay, I'm good. Right. So now that we have seen more or less what is in the light path, so it's not super complicated, there's not tons of things of them. I want to tell you that the world of light microscopy is divided in two only. And so that is a simplification, but it's actually, yeah, it works. So there are microscopes that have a camera, and these are all microscopes except point confocals. So wide field system, light sheets, and many more, they have a camera. And then we have microscopes without a camera, and these are the single point confocals. Okay, so there's a vast difference between a single point confocal and all the other types of microscopes. And this is what we are going to talk about. And this is why this is for single point confocals and this is for cameras. So let's have a definition here as well. In this course, we're going to talk about single point confocal. And this is what everybody calls a confocal, or sometimes point scanning confocal, or sometimes laser scanning microscope, like LSM, like this is what Zeiss calls them, okay? So all these things in this course, we're going to talk about, we're going to call them single point confocals. And the reason is that I want to differentiate this from multi-point confocal that I'll talk about later. And then when we say wide field in this course, we're talking about a system with a defocused illumination. So single point confocals, they have a focused illumination, and wide field, they have a defocused illumination. And for this, that's what people call a standard microscope. Sometimes people say fluorescence microscope or even an epifluorescence microscope, and that is not logical at all because, sorry, but a confocal is also a fluorescent microscope, or sometimes a normal microscope or a stupid microscope. So no, this is a wide field microscope. So from now on, these type of microscope which have a defocused illumination, we call them wide field, and then all the confocals we call single point confocal. Okay. So we talk about the same language here during the course. And so it's very simple in some ways. You need to recognize what a microscope is when you look at it. You need to check if it has a camera or not. And, oops, yes. So now I want to start this. And to start, we're going to talk about the architecture of the microscope body, the microscope stand, the one that has the stage. And also we're going to talk about transmitted light versus fluorescence. And so this is what we are going to talk about during these 3 little lectures I have this morning. In this first one, we're talking about these points, and then afterwards we'll talk about the next points. So now I'm going to talk about upright, inverted, and flat architectures. So here we have an upright microscope, and here we have an inverted microscope. And you can see here the stage. This is where we put our sample. So here is the stage. And in an upright microscope, the objectives are above the stage. So we have several objectives, but when we image, we only use one. Okay, we can change, but when we're imaging, we're only using one, but we have the choice of several objectives. It's the same thing in the inverted microscope. We have here an objective turret, but with several objectives, where we use only one when we image. Okay, so we have the stage, and the objectives are below the stage for an inverted microscope. They are above the stage for an upright microscope. This is the way we recognize upright and inverted microscopes. Now, the thing is that there is a condenser on the microscope, and the condenser is always opposite the objectives compared to the stage. Okay, so you can see here in this upright microscope, the condenser is below, and here in this inverted microscope, the condenser is at the top. So what is a flat? When we have a light sheet microscope, which uses often several objectives while we're imaging instead of using only one. So in this light sheet microscope uses 2 objectives to image. This one is upright because we have the stage here and the objectives are above. Okay. In a light sheet microscope, there is no way to image transmitted light microscopy, so we don't have a condenser under the stage. So this is an upright light sheet, but it is possible to get a flat light sheet like this one. And in this case, the objectives are at the same plane as the sample. So you can see in this microscope, this is the stage. This is where you put your sample. And if we look from the top and we open all these little boxes, this is what we see. So the sample is going to be here on the stage, and you see that the objectives are all in the same plane as the sample. And so this is a flat architecture. And in this light sheet system, we have 4 objectives at the same time to image. So light sheets are clearly have a very different architecture. But some of you may have an upright microscope, and some of you may have an inverted microscope. So I want to tell you why we use one or the other. So this is the stage. This is the objective. So this is an upright microscope. The condenser is here below. And this is a little bit historical first. The first microscopes were upright. And so I think for quite a long time, the microscope stayed upright because of that. But also, upright microscopes are very good when you have a thick sample and you want a sample that cannot be flipped. For example, a sample that has liquid on top of it, or an animal that is alive and you're doing some physiology on it. And if you flip the sample, it's not physiological at all, like a mouse. You don't put it on its back, right? So it's also very useful when you need quite a lot of space here and you still need the condenser. Condenser is very big in general, so it's convenient to have lots of pipettes to have the objective above. So when you use when you image in intravital, it's not always the case that you image with an upright microscope, but quite often, if you're imaging here at the top, you don't want to flip the animal or in a cranial window. So this is the reason why we can use an upright microscope. The problem with an upright microscope is because the objective is on the same side as the sample. If you move the stage, the objective will bump into the sample. So there's very little stage movements for an upright microscope. Now, this is an inverted microscope. We can see the stage here, and the objective is below the stage. So that's the definition of an upright microscope. And as always, the condenser is on the other side. And it's much easier because now the objective is not on the same side at the stage as the sample. So we can use, for example, CO2 chambers and still move the stage without bumping into the objective. So very convenient. So anytime you want to combine large stage movements with high resolution, like for example, you can use like an oil objective and everything, you definitely want to have an inverted microscope. So for that reason, because of this convenience, most high-end microscopes for most applications are inverted, but except for high-end microscopes with a very specialized applications that I have mentioned before, they can be upright. So for fixed samples, if you have a fixed sample between slide and cover slip, it doesn't matter at all if you use an upright or an inverted microscope. You will get exactly the same difference because your objective is not going to bump into the sample since it's very thin and flat. So now here is a little bit of a list of the things that are common and different between an upright and an inverted microscope. And these are the things that are the same. So on an upright or inverted microscope, we can use them as a wide field on a wide field system, a confocal system, light sheet system. We can image fluorescence and transmission. We can have full automation of XY, so the stage moving and then doing Z stacks and also changing colors. So this is the same on both. And we can image live samples or fixed samples, and we can do some advanced techniques like FRAP and FLIM and all this stuff on both of them. However, when the upright microscope is best when imaging the top of thick samples that cannot be flipped, and the inverted microscope is best for everything else. Another disadvantage of an upright microscope is that we have no possibility to have a hardware autofocus, and we will talk about this later in the course, whereas here we can have that. So this is a little bit the difference between upright and inverted microscope. Now, if we look at the light path on the emission side. So what is the light path on the emission side? It's very simple. It's mostly where does the light go from the sample. So the sample in fluorescence is a light source. And so we have from the sample to the objective to the sensor. So in general, the emission light path is quite simple. So we can see in here our sample is this upright microscope. On the sample is here on the stage, and when we want to image, the light is going to go through the objective. So from the sample to the objective to here is a camera to the sensor. So it's a very simple light path. And then in here, and then in here on this inverted microscope is the same thing. We go from the stage to the objective and to this, which is a camera. So this is very simple. It's a tip that I give you now. The sensors are mostly on the same side as the objective because mostly the light path goes through the objective and then to the sensors. So these two are on the same side of the microscope. So here is the light path. We go either to the camera, so we image with the camera, or to the eyepiece. And then it's the same thing in here. We go from the sample to the objective, to the camera, or to the eyepiece. So this is the emission light path for these two systems. There's only one exception, and this is when we are imaging transmitted light with a single point confocal. So we're going to talk about how that works. So what about the excitation light path then? Where does the light go? Well, that's a little bit more complicated. So there's a difference between transmission and fluorescence, and there's a difference between confocal and wide field. So we're going to have a look at this now. But first, we're going to do a little quiz. So I would like you to go to Canvas and look at this microscope architecture. Stop share. No, not stop share. I want to click. Yes. So microscope architecture quiz, which is here. And then you do that. It will take no time. So is this microscope a